Hello everyone, my topic is types of antigen antibody reaction. Antigen antibody reaction brought about by the contact of antigen and antibody. This process of contact cannot be seen by the naked eye. But once contact is made, the AG-AB reaction leads to visible manifestations such as precipitation, agglutination, and cytolysis, etc. The antigen antibody reaction can be detected by the following techniques: precipitation, agglutination, cytolysis, complement fixation, flocculation, opsonization, immunofluorescence. So the first one is precipitation. The precipitation refers to an antigen antibody reaction between a soluble antigen and its antibody, resulting in the formation of insoluble precipitation. The antibody causing precipitation is called precipitin. Example: the reaction of antibody with BSA, bovine serum albumin. Mechanism of precipitation. Precipitation is due to the formation of antigen-antibody complex. The antigen is multivalent and the antibody is bivalent. As each antibody is a bivalent molecule, it can bridge to multivalent antigen molecule. This bridging leads to the formation of lattice, which forms the precipitate. When antigen and antibody are optimal concentration, the precipitation is complete and the large lattice is formed. So the application of precipitation reaction. The precipitation test is used to find out the amount of antibody present in the serum of a immunized animal. VDRL test is based on AG-AB reaction. It is a test for syphilis. Alpha fetoprotein of liver tumor can be detected. Serum proteins and urine proteins can be detected. Hepatitis B antigen can be identified. Systemic lupus erythematosus can be detected by the precipitation test. Precipitation reaction is basic principle of the following techniques: single immunodiffusion, immunoelectrophoresis, double immunodiffusion, rocket immunodiffusion, radial immunodiffusion. The next topic, agglutination. Agglutination is a antigen antibody reaction where the antibody of serum causes the cellular antigens to adhere to one another to form clumps. It is the clumping of the particular antigens and its antibody. The antibodies that causes agglutination are called agglutinins, and the particulate the antigens aggregated are called agglutinogens. The particulate antigen include bacteria, virus, RBC, platelets, lymphocytes, etc. When red blood cells are agglutinated, the reaction is called heme agglutination. When bacterial cells are agglutinated, the agglutination is called bacterial agglutination. Mechanism of agglutination. Agglutination is brought about by the linking of antigen and antibodies. As most of the antibodies are bivalent, an antibody can link to adjacent antigens. The IgM antibody is multivalent and it contains 5 or 10 combining sites. Hence, it can link more number of antigen. Hence, IgM antibody has the capacity to make clumps more effectively with lesser number of molecules than that of IgG antibody molecule. The univalent antibodies cannot form clumps or lattice, and hence agglutination will not occur. Agglutination test. Agglutination test refers to the examination of clump formation where particulate antigen and its antibodies are combined. ABO blood group typing. The typing of blood for ABO groups or anti-RH groups involves an agglutination reaction. For typing blood, a drop of blood sample is mixed with a drop of antiserum A, and another drop of blood sample is mixed with a drop of antiserum B on a glass slide, and anti A plus A blood, anti B plus blood samples group A. If blood sample is clumped with anti-serum A, the sample belongs to. If the sample is clumped with the anti-serum A, the sample belongs to. If the sample is clumped with the anti-serum B, the sample belongs to B. If the sample is clumped with both antiserum A and antiserum B, the blood sample belongs to AB. If there is no agglutination, the blood sample belongs to group O. Applications. The following are the application of agglutination test: ABA blood grouping, RH blood grouping, Weidel test for typhoid, Well-Felix test for typhus, Coombs test for identification anti-RH antibodies, Brucella agglutination test for brucellosis, Leptospira agglutination test for leptospirosis, cold agglutination test for pneumonia, malaria, trypanosomiasis. The MSCs make holes of the plasma membrane of the pathogen. The cells contain the pathogen flowed. This leads to the lysis of the pathogen. Next one is complement fixation. The binding of complement to antigen-antibody complex is called complement fixation. When the antibodies bind with the pathogen, the antigen-antibody complex is formed. The antigen-antibody complex activates the complement 1, C1. The C1 binds to the with the antigen-antibody complex. This is called complement fixation. The binding of C1 to the antigen-antibody complex causes a cascade of complement activation. The byproduct of the complement are deposited on the pathogen to form MSCs, membrane attack complex. The next one is opponization. Opponization is the process by which antibodies bind to the pathogen and the pathogen is marked for killing by phagocytes. The marking substance is called opsonin. Opsonin is a molecule that targets antigen and stimulates the immune response. Molecules that act as a binding enhancer for the process of phagocytosis are called opsonins. The mechanism of opponization. The opsonin binds to the receptor of the pathogen. This results in the formation of antigen-opsonin complex. The far portion of the opson binds to the spin comb with the pathogen. When antigen-opsonin complex is formed, the antigen is opsonized. The opsonized antigen is marked for destruction. The opsonized antigen is killing in 3 ways: phagocytosis, complement fixation, complement activation, and antibody dependent cellular cytotoxicity. Opsonins coat the negatively charged molecules present on the surface membrane of antigens. The molecules that activate the complement system are called opsonins. Opsonin can also refer to molecules that target a cell for destruction through the action of natural killer cells. IgM type of antibody is extremely efficient in activating complement. Hence, IgM is considered as an opsonin. Examples of opsonin: antibodies such as IgM if complements such as C3b, C4b, C3bi, etc. Mannose binding lectin in opponization. The antibody combines with the surface of antigen of bacteria to form antigen-antibody complex. The antigen-antibody complex activates. The first type is phagocytosis. The opponized pathogen attracts phagocytosis. The Fc portion of opsonin binds to the Fc receptor of phagocyte. This binding stimulates the phagocytosis. The pathogen is swallowed and degraded into fragments. The രണ്ടാമത്തെ complement Activation. The antigen opsonine complex activates the complement system. The byproducts of complement activation are attached on the pathogen to form MAX. And the MAX makes the holes of the plasma membrane of pathogen. The fluids flow hand of the holes. This causes the lysis of the pathogen. The third one is antibody dependent cellular cytotoxicity. The antigen opsonine complex attract the immune defector cells such as NK also, monocytes and neutrophils, eosinophils, etc. The Fc domain of the opsonine binds to the Fc receptor of the immune effector cells. This binding triggers the release of lysis products such as perforin and knee-bound immune effector cells. The lysis products lyse the pathogen. The topic is over. Thanks to everyone.